Okay. Well, thank you very much for joining us to this another session of our Public Journal Club in Immunology. And today we're going to discuss the paper entitled Cytidine deaminase 2 is required for VLRB2 antibody gene assembly in Lampreys. So my name is Eugenio Contreras. I am a current, I'm a former PhD student at I'm current PhD student at National Autonomous University of Mexico. And well, today we're going to check this paper that was done by the Thomas Boehm lab at the Max Planck Institute of Immunology and Epigenetics. And this work was mainly done by Ryo Morimoto and Connor Meara. And also it was important, the work from the National Institute of Agricultural Research in France, where they did all the CRISPR mutants. So I'm gonna start asking you why you think this publication is important in the area of immunology. Yeah, this is a really good point, and also because this is the first time that they did a CRISPR in lamprey, in this lamprey that is a species that live in the river, and they finally proved this hypothesis that this gene cytidine deaminase 2 it was important for VLRB antibody assembly. Okay, so as a matter of introduction, we know that vertebrates, we have a common ancestor of the vertebrate. I'm sorry, I'm gonna put my here. No. Here. So we have a common ancestor of vertebrates, and you have two groups: the agnata or jawless, or natostomata or jaw vertebrates. Agnata includes hagfishes and lampreys, and natostomates includes cartilaginous fishes and other vertebrates, including mammals. Okay, and when we compare the difference between these immune system invertebrates, what we can see that there are several features that are conserved during evolution. For instance, we know that the gut-associated lymphoid tissue is conserved in all vertebrates. These bone marrow equivalent, mainly a site of hematopoiesis, is conserved between vertebrates. And for example, the thymus is another organ that is conserved in all vertebrates. But for example, lymph nodes only appears in mammals and birds, and spleen, I think they don't. We start seeing spleen early in lampreys in cartilaginous fishes. Okay. Also, I want to tell you that if you you see that if you want to add some comments, you are welcome to do it. Okay. So when one of the major novelties of vertebrates was actually the lymphocyte-based adaptive immunity. And for example, the natostomates or the jaw vertebrates have these novelties, such as TCR and BCR structures that are expressed on these lymphocytes. And these genes are rearranged by this enzyme RAG1 and RAG2. And the process of activation of these TCR and BCR receptors in lymphocytes is dependent on partially of MHC class molecules. In comparison, the agnata or the jawless vertebrates, including hagfishes and lampreys, they lack these genes, and they have other genes that resembles the function of lymphocytes. For instance, we know that we can find lymphocyte-like cells, and we can split into groups. The first group will be the ones that are generated in the thymus, and we could find BLRC lymphocytes, which resembles like the gamma delta T cells in natostomates, we also have this VLRB positive lymphocyte, which resembles alpha beta T cells in natostomates. And well, these lymphocytes are characterized by the expression of VLRC or VLRA and are developed in the thymus. Their development is dependent of this gene that is called CD81. Okay. And in the other hand, we have the lymphocytes that express VLRB, and these cells are generated in a structure in the lampreys called tiflosol and the kidney, which are mostly hematopoietic sites of generation of cells. And these VLRB cells are characterized for the production of some antibodies, or what they call agglutinins, in a pentameric form. So they resemble the B cell function. So for instance, we see that these jawless vertebrates, even though they don't have TCR, BCR, or B or T cells, they have a sort of similar cells in those animals. Okay, so I'm gonna start talking about VLRs, and VLRs are the variable lymphocyte receptor of these jawless vertebrates, and they are characterized by the expression of a leucine-rich repeat that we're seeing here. So this is the crystallography structure of these VLRB, VLRA, and VLRC. And it is important to notice that VLRB is the only receptor that can be secreted. So in this way, we have a sort of antibody-like structure that will protect the animal. Okay, and well, this is the gene that is assembled after expression. This is the crystallography structure, and I want to mention that this LRCCT is the sequence, or the sequence. I'm sorry, that is gonna bind the antigen. That's gonna bind the antigen. Okay. So when we do zoom into the genome of the lamprey, we can see that these VLRI, VLRB, and VLRC genes are located within the genome, but particularly are in order to be expressed and correctly express the receptor, the gene has to be processed. How is this process? In a process that is called a gene conversion-like process. So in figure B, we see a sort of cartoon where we can find this sequence that is conserved, and flanking this sequence, we have other LRR repeats that needs to be assembled. I'm sorry, in order to produce a correct expression of the gene. So particularly VLRB can be assembled in two fashions: in a 5' to 3' assembly, or a 3' to 5' assembly. This process is controlled, or is hypothesized to be controlled by a gene called CD82, which it has the ability to assemble these genes, and it has homology with a gene called 8, or activation-induced cytidine deaminase, which we know is a gene really important for the expression of TCR and BCR in jaw vertebrates. So the question, or the main question of this work is CD82 required for VLRB assembly in lamprey? So I'm going to start now talking a little bit more of the experimental procedures, but I want to ask if you want to add something to the introduction, guys. No? Okay, perfect. So what they did first was to one of the main goals of this work, as I mentioned before, it was to determine if if CD82 was required for VLRB assembly, and so for this did they established this protocol for long-term maintenance in vitro fertilized lamprey larvae, where they took these eggs from Lampetra planeri, and they do in vitro fertilization. They after they put the eggs in vitro in petri dishes. I'm sorry, for 5 weeks, and after that they put the larvae into tanks for their natural cycle of life. Okay, so after they did this protocol, they wanted to know how was the expression kinetics of VLRB in lamprey during development of lamprey. So and they use primers for detection of the I think the LRCCT domain of the gene. And what it was important for the authors is that they noticed that they see expression of VLRB until week 6. And now after week 14, they see an enrichment of the expression. But the important here is that we see expression at week 6 after fertilization. And when they look for the assembly of the gene, so we have the one thing is the expression of the gene, and the other thing is the assembly of the gene. They see that even though we see expression of the gene at week 6, we start seeing an assembly of the gene only until week 10, and we see clearly here. And when we compare, or when the authors compared the assembly of VLR-A and VLR-C genes, they see that the assembly of the genes are delayed until week 12. And we see it clearly on figure D when we see a kinetics of the expression of VLRB, and we see that we first is the expression and the assembly of VLRB, and afterwards we are gonna see an expression of VLR-A and VLR-C genes. Okay, so we conclude with this figure that VLRB assembly precedes VLR-A and VLR-C. Okay, so the next question, and what they did was they wanted to characterize the VLRB gene repertoire, and for this, what they did was to take 10 to 16 week-old larvae. They also did with 2 years larvae, and what they did was put all the animal into cold ice or in trisol for purification of RNA to look for the assembly of VLRB. So once again, they use primers or for the detection of LRCCT, which is the sequence that is important for antigen recognition, and is mostly variable in these VLRB genes receptors. So what they did, and the results that they have is that when they compare the VLRB gene repertoire of larvae ranging from 10 to 2-year-old animal, and when we look at the loop length or the amino acid length of the sequence that is here at the LRCCT, we found a bimodal distribution where we see an enrichment of the sequence of the frequency of the 19 amino acids. Look at here, 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 and here. And also an enrichment of 26 and 27 amino acid sequence, right? Look here, 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 here. So it seems that this bimodal distribution is conserved during development of the animal. And what was really relevant, they sequenced they have now they identify the sequences of these loop lengths. And what is relevant for their studies is that they found a two conserved sequence that is maintained during development until 2 years old animal, and this is somehow they hypothesize that this is sequence is important for VLRB recognition of the antigens. So we have the frequency and the characterization of the sequence, and they looking at the frequency results. So they wanted to know if the result that they were seeing was dependent on the copy numbers of the LRCCT. So for instance, if we are seeing this distribution, we have two options. The first one is that we have an enrichment of 19 or 26 or 27 sequence in the genome, or is independent of the sequence. So they had the genome of the lamprey, and they did this correlation between the frequency of the usage of the frequency of the usage of the repertoire of the receptor, and comparing with the numbers of LRCC cassettes in the animal. And they don't found any correlation between LRCCT numbers and the frequency of usage. So this means that what are they looking here is not dependent on the numbers of the LRCCT cassettes, and it has to be other mechanisms that is unknown. And after they have this graph, which I don't know if someone could help me to interpret. According to the text, this is they say with this text that the VLRB assemblies follows a power law distribution, which means there's a preferential use of short LRCC cassettes. This is a week-old and a 2-year-old animal. In red, we see the 19 amino acid sequence, but I don't know if you guys could help me to interpret this figure, because I have no idea on how to interpret this data. Anyone? Or we could ask Morimoto at the end of the session.
I mean, is that there is non-specific preferences in the usage of the short amino acid sequences in the repertoire of VLRBs. I thought that is the main idea of D and E figures. So if anyone on the meet has a different idea, it could be good now to explain that figures.
I don't know if in this figure the rank is referred to the statistical value, or in which rank is the difference between the maximum and minimum value. And in this case, I suppose that it is like Aranzazu says that there are a more frequency of cells of lampreys with a lower difference in their repertoire of VLRB assemblies. I think, but I don't know if my idea is correct.
Yeah, I think it's correct what Aranzazu and you said, but yes, it was the first time I see this power law distribution graph, so I was really confused with this whole idea. But yes, the idea is clear that there is a preferential use of this 19 amino acid sequence in the repertoire of the VLRB. Thank you so much. And finally, they compare the usage of the VLRB repertoire between a young larva and a wild caught animal of 2-year-old, and we see a nicely correlation. So it seems that I mean, my conclusion that I thought after reading this is that the result is not dependent of any laboratory condition, but in fact, is also found in the wild. Okay. So, well, with this, the authors conclude. Yes. Yeah, it might be a possible explanation, which I think is really a good idea, because yes, I think that this animal could have been exposed to any thousand of antigens compared with the young larva. So yes, I think it's a really good point. We could, yes, at the end we could ask Morimoto at the end of the session. Thank you, Diego. So with this, they have this conclusion, which I want also to ask Morimoto, because they conclude that LRC cassettes with short invariant loops mark a population of VLRB sequences that are either tuned to recognition antigenic determinants of limited structural diversity or favor binding. I don't know very much about VLRB, so, but I don't know why or how they could get to this conclusion. I mean, the limited structural diversity or favor binding. This is two conclusions that I honestly don't have any idea of how they get to that. I mean, I guess for the limited structural diversity, a limited number of amino acids could, in fact, promote a limited diversity, but think that they only found two sequences here, two conserved sequences here. So,
well, I guess in order to understand this, how VLRB repertoire is working, I guess it's good to compare with what happened in T and B cell receptors, because you can figure out how these receptors can be very has very diverse regions that they mentioned before, called I can remember LCCR. No, it's
LRCCT.
No, it's LRRB. That is the most variable section of the VLRB. So they assume that the region of LRRB on the VLRB it maybe contribute more than the LRRCT regions in the whole receptor, and can contribute in the recognition of the antigens.
Yes, it was actually, that is one of their hypotheses, the favor bindings, that it favor bindings having these short invariant loops. Well, okay, so after this figure, they now look for I think the main reason of this paper was to characterize or to make crispants of CD2. So first, after doing the crispants, they do a kinetic of expression of CD2 during larva development. And what is really interesting that at week 10, we see an enrichment of this CD2 expression, which it correlates really nicely with the presence of the VLRB2 assembly gene. So it's really a nice correlation. And after that, they did this crispants targeting exon 1 of CD2. Here we have the NGG sequence and the guide RNA targeting sequence. So they proved in vitro that their guide RNA was working. And this is where the primers for detection of TDA2. And then, well, they make these crispants and yes. Yeah, thank you. Thank you very much. Yeah, thank you. So this was the way they did the crispants, and here we have the phenotypification of the animals. We have the control animals where they have a complete a normal gene, so it's a wild type. And when they look at the crispants, we have that according to the text that they have most of the animals have biallelic deleterious mutation. These are animals that are 26 weeks old. Some of them, they have no mutation at all. We see it here. But most of them, they have biallelic deleterious mutations. So, for example, after growing the animals, they sequence the genome of the region, and we see here, we have here 3 examples, animal 32, 31, and number 30, that they have different, they have mosaicism in the animals. We see in red here in the asterisk is a mutation that is stop codon mutation, and here they have the deletion of some genes of some amino acids. In this animal, we have a deletion of amino acids, and here we have a frameshift mutation that causes an altered sequence with no functionality at all. We see it here and also here. Okay, so we have different type of mutation, different type of modifications using CRISPR, but most of them induces modification in the exon 1, and most of them interfere with the catalytic activity of CD8-2. And here, for example, they also look at if we have the mutations, we have then we expect to have no assembly at all of VLRB. And when they look at the assembly of VLRB, these are some animals. We see that, for example, animal 8 and animal 11, they still have a VLRB2 assembly. So when they look at what could happen during these CRISPR mutants, they found that, in fact, for example, animal 11, we started seeing mutations only after the second cell division in the 4th cell state. So in this way, the animal could still have a wild-type form of the gene that would induce at the end a complete VLRB gene assembly. Look with here, and you see here enrichment of mutation. And in the other way around, we can see here that we have some deleterious mutations starting at 1 cell stage, and then afterwards 2 and 4 cell stage, we still have gains of mutations using the crispants. Even though we have mutations here, we still have a VLRB2 gene assembly process. So here they conclude that even the crispants is really the CRISPR method is really how to say it, efficient, but we have some caveats during the process. So they have to check for the process. So, okay, after this, they have the conclusion of this figure is that they make a CD2 Lamprey crispants. So now they look for what happened if you don't have a CD2 functional gene. So here we have Innate, and we are looking at the expression of the VLRB assembled gene. So we have these crispants, and most of them they lack VLRB gene assembly. We have some animals that, as we have previously shown before, they have some mutations that are incorrect mutations that will still have a VLRB function, but most of the crispants don't have VLRB assembly. And what is important here, and I think this is one of the key results of the paper, is that you don't have assembly, but you still have gene expression. So CD8-2 is important for gene assembly, but not for gene expression, okay? And in particular here, I think I'm really excited with this figure, because when we look at the VLR-A and VLR-C gene assembly, Remember that these genes, this one is for alpha beta T cells, like cells, and this is for gamma delta T like cells. So this is the killer experiment where they show that CD8+ is only required for VLRB2 assembly and not for VLR or VLRC assembly. And this was they see this phenotype in 25 of 29 animals. And okay. And finally, the physiological, let's say, a role of CD8+ now in the animal. Here we have some histological images of the tiflosol here in T, which we know that in larva, the tiflosol is the anatomical site of hematopoiesis and also the kidney. So we have the control map, we see cellularity, and when we look at the CD8+ crispant, we look at the tiflosol or the kidney, we see a hypocellularity image, a lack of infiltration in these tissues. So what it means that maybe or perhaps we don't see any VLRB like VLRB cells in these tissues. And when they do this in situ hybridization using proofs against VLRB2 assembled gene, you have the control. We have a signal here in the tiflosol and in the kidney, but in the crispant, we don't see any positive signals in the tiflosol or in the kidney. And this is the result, looking at different crispants or control animals. We don't see any signal whatsoever in the mutant comparing with the control. What is also important, we don't see any inflammation whatsoever in the animal, and we don't see any difference between. They don't show the data, but they put it in the text that we don't see any difference in the thymus of the lamprey, and the infiltration of the thymus of the lamprey. So with this, we have reached to this conclusion that CD2A is required for VLRB assembly and survival of B-like cells in lamprey. So even though this, well, in fact, this is the conclusion of the paper, it seems simple, but in the other way, it's kind of complex, this whole methodology, and what the opportunity to make crispants in the lamprey now could open many possibilities of proving some of their hypotheses. For example, blocking or eliminating CD1, or to do some allograft experiments with this crispant. So it's really, really important and nice data that they have reached during this hard work. So thank you very much for your attention. And do you guys have any question? We will still have to wait to Morimoto until 10 o'clock, where he's gonna join us.
I have a question. So they found that this protein CD2A regulates this like the rearrangement, right? But they don't have a direct evidence that this is direct, right? So you have the mutant, but you don't know if this is an indirect effect of this particular protein, because it's reported to have other phenotypes, this protein itself, in other species. So I'm wondering if if this is a true evidence.
Well, I must say that I think yes, because we don't see any VLRB assembled form of the gene in the crispants. But yes, I don't know. I mean, I don't know if they have done this in vitro, where they put the protein and the gene, and if they could in vitro do the assembly. I don't know, but it's a really good question. Yes. Yes.
Yeah.
Work. Yes. Yes, one of the interesting thing, and I don't know if you guys know if happens the same with uh, uh, mammals, for example. What happened with when you see this time point or kinetics? Do you guys know if in upper vertebrates happen the same? Like B cells are produced before T cells. I don't know if this something that is conserved during vertebrate immune evolution. I don't know, guys, if you have any idea of this. Yes, I don't know, because when we look at the anatomy of the immune system of the lamprey, they don't have lymph nodes. So what is the I mean, I'm thinking of, if you still have a constant production of B-like cells or T cells, afterwards, where are they going to migrate? I mean, where's the niche that they could fill? So maybe, as you say, I don't know if there's a constant production of these cells, or it's maybe a time window. I don't know. It's a really good idea. Okay, so
I think it's also interesting that what happened with that cells that are in the crispans in the kidney and in the another tissue. I can remember the name. So
Tiflozoid?
well, huh? yeah, and that tissue there are also other cells. What kind of cells are those cells? Because you can avoid that cells, and I can stop thinking about what happened when you delete the expression of RAC in mice or in another vertebrate. You avoid also the other kind of T cells like ILCs or etc. So I was thinking about what this is a less sophisticated mechanism that involves only one molecule, CD8-2. Meanwhile, in mammals, you have a wide a wide spectrum of molecules that are regulating this the development of T cells and B cells. So I think it could be possible that this mechanism, these cells could be homologous systems of adaptive immunity, but also we have to keep in mind that these cells don't have the ability to generate memory, cell memory or immune memory. So I don't know what do you think about that? What a kind of evolutive mechanisms that are favoring the no, they don't have memory.
the lamprey don't have memory.
No,
Oh, I didn't know that. Yeah, it's really a good point. I didn't know about that. What is really relevant for the adaptive immune response of early vertebrates is that, for example, we have at least upper vertebrates, they have RAC genes, and these two genes are used for the TCR, for the T and B cells, and in comparison with the jawless vertebrates, they require separate genes. So yeah.
It will be interesting to think about what signals in mammals, for example, are responsible for the induction of memory. I have not clear. You're working with this, or you have
Yeah.
an idea of
Yeah,
which
I guess
signal?
one sign of this of that is that lampreys lack of no of secondary immune tissues, so they don't have lymphoid nodes. So it could be an insight of why they don't develop memory.
But the typhus, for example, is not like a immune tissue, because they say that they comment that when they analyze the CRISPRs, they observed that they don't have B-like cells, but they have yellow cells. But I
Yeah,
don't.
yeah, but B, L, R, A, and C develop in the primitive thymus. That lamprey has a primitive thymus. So these cells, well, these T-like cells can develop in this primitive thymus. Meanwhile, B-like cells can migrate to different tissues to complete their development.
Right, really good discussion, guys. So let's wait. Let's wait to Morimoto if you have any questions. I'm gonna advertise our next speaker, which is we're gonna check this paper entitled "Program Disarming of the Neutrophil Proteome Reduces the Magnitude of Inflammation." This discussion will be led by Emmanuel Flores. And the final discussion, we have really exciting news. We have Andrés Hidalgo, which is the last author, and we have Jose Adrover, which is the first author. So this is really a good opportunity, not only to ask the first, but also the PI. So we're really excited. Please share, and hope you can join us next week. So.
Eugenio, maybe they join us in the start of the paper presentation.
What?
Maybe they join us at the start of the presentation.
Okay.
Miguel Hidalgo and his student.
Yeah. Yeah, Hidalgo said me that.
At the
start.
At the start.
Maybe they open the possibility.
So
but
don't share the link.
It's not sure.
Until we end the session.
Yes.
So we can have time.
Yes.
Well, now what? Yeah.
Oh, yeah.
So we have a Twitter. Our
Twitter is you can find us on Twitter as Public Journal C, just C, Public Journal, and on Instagram you can find us like
Public Journal
Club, and on Instagram you can find us also as the same username, Public Journal Club.
Well, if any one of the here present are interested in joining us as formal members and leading a paper discussion in the future, we'll just say it.
So just wait a couple of minutes. Okay, guys, did you like the paper?
I have a question. Someone in the meeting know which are the signals to induce memory cytokine? Who induce a memory phenotype in T cells and B cells?
Yeah, that's sorry. You can also ask in the chat, guys. So if you want to ask on the chat, and so then one of us can ask your questions to Morimoto. So
Eugenio,
Yes.
you are gonna present Morimoto when he's present. When
Yes. Yes. Yeah,
well,
because imagine I present it now and then we wait 10 minutes until he arrives.
well, you're right.
Okay. Is there a time window when lampreys are able to produce B and T cells? No, I don't know. Okay.
Diego, you're absolutely no shy.
Yeah, this is a really good idea.
Before asking something to Morimoto, remember to introduce yourself with a little bit sentence, please. What is your name? Where do you work? What do you do in your labs?
Your hobbies.
Your
No, hobbies, no. Hobbies.
Your Netflix favorite.
But like before you ask him anything, or I mean, in general, just like presenting itself like an anonymous alcoholic, yeah, I'm this person, I have this problem.
like
Okay, perfect. No,
I guess no one here actually works with evolution or lamprey, so
no. I found it not. I probably work in a total different area, so that's why I was interested in immunology and all your presentations.
Oh, thank you. Kevin Galindo, anyone? A ver. Yes. Yeah, and it's really curious of what happened with VLRs, because when we look at the jaw vertebrates, the B and T cells, in order to be activated, they have to have MHC class molecules, and we have Morimoto right now. Okay. Okay. So thank you, Dr. Morimoto. I'm gonna start a brief presentation of your work. So let me just 1 second. Okay. So first of all, I wanna thank Dr. Morimoto for joining us today in this section of Public Journal Club in Immunology. And Dr. Morimoto was born and raised in Tokyo, in Japan. He got the medical degree from the University of Tokyo, where he where he got the degree in medicine. After that, he moved to research, studying lipid biochemistry, where he studied phospholipid metabolism and production of inflammatory lipid mediators. After that, he moved to Germany to work with the Boehm lab since 2017, where he, as you all know, he works with Lampreys, and he is really interested in evolution and particularly in the immunological aspect of evolution, I think. Thank you very much, Dr. Morimoto, for joining us today. And we have a couple of questions. Guys, if you could ask Morimoto. Doctor, I don't think we could hear your microphone. No, we don't. Yeah.
Is it okay?
Yes. Yes.
Ah, great. So
Great.
thank you so much for your kind invitation, and thank you so much for choosing my paper for your very nice International Journal Club. I'm so happy to hear from you, and I'm very happy to take any questions. So please let me know how you discussed about my paper, or any kind of question is very welcome.
So,
hello. Sure. Mm-hmm. Mm-hmm. Thank you. Yeah. Mm-hmm. Yeah. So thank you very much. Yeah. So this is very important point. So this is a very fast advance in the lamprey rearing in the laboratory environment. So this is kind of, so the CDA2 result itself is very interesting. But before that, this the system to keep lampreys in the laboratory environment was a huge advantage that we created. So now we confirmed that this system can maintain the mutant animals. So of course we are now aiming at more physiological or pathophysiological experiments. So for example, there are some infection experiment model, like injecting some germs or some, for example, poly-IC or something like part of the germs or the pathogens. So we are now thinking about how to proceed these experiments and what to expect from them. So basically, this animal seems to survive quite a long time, even without VLRB. So which must be very important arm of the adaptive immune system, but somehow in the relatively clean laboratory environment, they can survive for quite a long time. Now we are kind of questioning what is the kind of orchestration between adaptive immunity and innate immunity in the lampreys. So your question is really pointing very important aspect of our project, and the short answer is kind of yes, and we're trying to make the very good condition for that. Thank you.
So I'm gonna have we have this question from Kevin Galindo.
Sure.
Kevin, you wanna say the question? I will say. Okay, I think I will say it. So Kevin asked if does anybody know if exists any functional difference between the lengths of loop sequence in VLRB assemblies?
The loop sequences. Sorry, I didn't hear you very well this time. For the loop sequence, could
Yes,
you please?
Is there any functional difference between the lengths of loop sequences?
Sure. So this is something we are just surprised from our analysis. So from the structural analysis, this loop region is already predicted to have a very important role to recognize the antigens. But of course, there's some information that this loop length is kind of different, and according to the length, the proto-region to the this groove in the VLRB receptor is different. So the longer the proto-region, also longer. So maybe this would be the kind of this. Sorry. So this would be explain the function difference or difference of the recognition of the antigens by the VLRBs, but we are not so sure. And what was important and interesting to us from the result is this short loop. If you look at our VLRB analysis part, the short VLRs, like, yes, this 19 amino acid one is kind of very stable. The other loop lengths are kind of distributed in as a single mountain. But for the very short one, 19 amino acid loop lengths, it's kind of consistent among all the ages. So I would guess these very short loop lengths might explain certain innate type or more like a pattern recognition type receptors. But this is just a mere guess. So we have to clarify this with the experiments. So I would say there must be certain difference in the functionality of the loop length, but we are still on the way to clarify this. So maybe with this CRISPR-Cas9 system, we can target these short VLRB specifically. Then maybe we can see some difference of the reaction of the animals to maybe infection models, or maybe they might develop autoimmunity or something like that. So we are totally at the starting point of this question. So this is a really good point. Thank you very much.
Hi, Dr. Morimoto. My name is Aaron Susu, and
Hello.
I'm ready to start my PhD Germany also. So
Oh, really?
regarding so regarding to the VLRB repertory characterization during the you use amino acid frequency on the loop length, singularity of amino acid on the loop length, and the average of usage as indicator of repertory diversity. So, do you have any idea of why the average on the usage on the usage in the loop length is the unique indicator that decreases according to the Lamprey aging?
This is a very interesting point too. So, this is So this is really important question, and this is something we get. We also struggling from the current results. So there are some specific pattern. Maybe this VLR cassettes and this LRRCT cassettes. So there must be some preference. Maybe it depends on the how to say this catalytic activity of CDA2, or maybe the location in the genome or something. So there is some bias between certain this variable cassette and LRRCT, this last motif that contains loop length. But for this difference among or over the course of the development, it's quite difficult to explain, I have to say. Maybe this, for example, if you are looking at the same display, the loop length 29 is kind of growing according to the age. So this might explain certain like exposure to the antigen, or so still almost everything is kind of open question. So yeah, this is really good point, but we don't have very clear answer yet. But I would say this is a result from more antigen exposure, and maybe there could be certain like education, like our T cells. There could be some education system to avoid autoimmunity or something. Then certain lengths or certain structure is more preferred and more amplified over the age. So this could be relatively rational assumption for now.
Okay, so thank you. I will stay
No
to know
problem.
your
Yeah,
work.
thank you very much for your kind of question.
We have
Hello,
another-
I'm
Go ahead. Go ahead.
Hi, I'm Carlos. I'm
Carlos,
also doing
hi.
I'm also doing my PhD in Germany in the same
Yeah.
institute
Yeah.
as you. I have a question. So in one of the first parts of the article, you mentioned something about the variation that there's, and you explained the variation due to the genetic background of the sampling that you do. So I'm wondering if you have done a systematic analysis on this, like if you have in the variation, it's also associated with a particular phenotype and also associated with a particular genetic background. So have you kind of do an analysis of the genomes of all these different strains and try to find like some genes that could explain these differences?
So variation means the variation of the mutants, or sorry, I didn't get it very well. Could
Yeah,
you repeat the word? Yeah.
Yeah, in the paper you mentioned that there's a variation between the weeks in which the maturation. Yeah.
I see. So difference between the age.
Yeah.
I see. Yeah. So
you say that overall the onset of immune maturation is different.
Mm-hmm.
And you explain this by the genetic background.
Yeah. So
I
I see. So something a bit difficult about dealing with these species is that in the same river, there are 3 lamprey species, and it seems that there is some, how to say, hybrid between the species. And then usually this species is not very well fertilized to the next generation, but still some genes are kind of mixed. So this would give us our the variation that we saw. But at least for the VLRB assembly and the order of VLRB assembly and A or C assembly is kind of kept. This is very consistent, I can say. Does it answer your question?
Yes.
Great. Thank you.
So I got this question regarding this expression of VLRB time point. So I think I have two questions. The first one will be, what does drives the expression of VLRB at early time points, such as 6 weeks? 6 weeks. What is the signal that drives expression of VLRB before the VLR and VLRC genes? And if there any physiological relevance of expressing first the VLRB and later the VLR.
So this is also a very interesting question. So the development of lymphocytes in lampreys are also not so very well studied. It seems that T cell lineage expressing VLR-A or VLR-C develops the gill region of the lampreys. And maybe the VLRB species, VLRB expressing B lineage lymphocytes are developing either in kidney or tiflosol, as I showed in the later figure in the histology part. So this is also very interesting regarding to the evolutionary questions. So maybe VLRB type assembly has a bit more ancient roots in the development of the adaptive immune system of lampreys over the course of evolution. So maybe VLRB is a bit more coming earlier, and VLR-A or C comes a bit later. But for the physiological meaning, this is also a bit confusing to us, because usually T lineages in jawed vertebrates are kind of educated in the thymus. But in this system, it seems that VLRB just starts much earlier and VLR-A or C comes a bit later. Then how do they avoid autoimmunity? This is a very difficult question for us. So regarding this point, the very early VLRB that we observed, for example, like 10 weeks or something, could represent more innate portion or VLRB. So of course, this is located in the adaptive immunity, but still, I would guess these VLRB observed in the very early stage still have kind of innate or pre-selected feature that we observed in the this repertoire analysis. Maybe this very short 19 amino acid loop lengths might represent a bit more innate character, then they come a bit earlier. But we have to confirm this a bit more carefully. This is mere assumption. So we don't have clear answer, but we can still guess something. But still we are working on that too. So development of lymphocytes itself is very controversial. Does it answer your question?
Yes, thank you very much.
Thank you.
Well, I have a question.
Sure.
Oh, hi, I am Emmanuel. I'm an undergraduate student in Mexico. I'm working now in signal transduction area. And well, I have a question. Do you know if upon antigen recognition, CD2 will participate in class switch in B cells in lamprey?
So far, we don't see class switch type modification to the lamprey antibody. So this would be a bit different system. So once the VLRB is there, it seems that this VLRB is just like IgM. Maybe they have like a tetramer or pentamer, and then they secrete it as it is. And it seems that there's no class switch recombination so far. So this is the first answer. And since you are doing this signal transduction, and your question is very interesting because you said upon antigen recognition, so there could be, or there must be certain signal transduction.
Yeah,
and,
for sure.
and how do you say, the downstream of the VLRB signaling. But so far, we don't have any information on this signal transduction. So I have to say this field is still at a very early stage of the development. So we really don't know the very basic question, which is already known in our jawed vertebrate system. But from this lamprey system, of course, if we just compare the genomes, we can find like a similar, like a homolog type molecules in the lamprey genome. But the antigen receptors are so different between jawless vertebrate and the jawed vertebrate. So we don't know whether they are working in the same way in the jawed vertebrate or not. And so far, I think it's quite difficult to deal with in vitro experiment of lamprey lymphocytes. So we don't have so much information yet. So thank you very much for the question.
Thank you.
And I don't know if Enrique,
Yeah, please just take your time, and
yes,
I'm very happy to answer.
well, I do have another question. It's
Sure.
so I don't know if you have sequenced the microbiota of the lamprey, and I don't know if this microbiota could change within larva development and the appearance of the enrichment of some species of the microbiota will correlate with the expression of the VLRB genes.
Yeah, this is also a very interesting question. So, of course, there must be a lot of how do you say, the conversation between microbiota and lamprey immune cells. But so far, we haven't touched this question yet. And what we can speak about this microbiota thing is, at least we didn't have severe, for example, inflammatory phenotype from CD8 mutant animals. And then I kind of expected some autoimmune phenotype or something from these animals, because they don't have VLRB, and of course this would have certain impact on the how do you say, this arms race between the microbiota and lamprey immune system. But it seems that there wasn't a very prominent inflammation on the gut. The other point is, since these VLRB cells are located in this typhlosoll, which is the spleen equivalent of the lamprey, and this typhlosoll is the invagination to the gut. So of course, this organ must have huge effect on the microbiota, like a maintenance of microbiota. So this would be very interesting and future questions, but so far we haven't reached to that stage yet. Thank you.
Thank you, Doc.
So I'm
Sure.
me again.
Yeah, please.
So
Yeah.
do you know B-like cells are monoallelic, biallelic, and there is a possibility of BR A and C double positive B-like cells?
Okay, that's very important question. So at least so far, we from the very limited number of lymphocyte analysis, we don't have. So we saw monoallelic expression of the VLRBs and also VLRA or C. And so far, it's very clear that VLRB lineage and VLRA or C T-like cell lineages are completely different lineages. So there's no double positive for VLRB plus A or B plus C. But so VLRA or VLRC is a bit different story. It seems that some cells try to assemble VLRC, but if it fails for 2 alleles, then it proceeds to VLRA assemblies. So there are some evidence for that. So at least B-like cell and T-like cell lineage is completely different, but within the T-like cell lineages, VLRA and VLRC is the lineage specification for VLRA and VLRC is not yet very clear. So this is the information so far we have, and we also would like to know what is the real difference between VLRA or VLRC. And additionally, so far we haven't observed VLRA, VLRC double positive cells. So there are some evidence of that, the attempt to assemble VLRC and VLRA in a certain order, but there's no cells expressing both at the same time as a functional receptor. So this is the answer for your question. Thank you.
Yeah, thank you.
So we have another question from our chat. So
thank you. Yeah.
From Victor. Victor is a genomic science student, and he asked, they observed that the overall immune system maturation varied by several weeks in the specimens. They mentioned that maybe because of genetic background. This variability happens in other species across evolution or happen in a strict time span manner.
Okay, they observed the overall immune system maturation for several weeks in their specimens, and this variety occurs in other species. Yeah, so this is a bit confusing point for us, and this is super multi-factor thing, and of course we just observe the variation in our laboratory system to rare Lampreys for a long time. The first, we have to say that this rearing system is still not so stable compared to, for example, zebrafish environment or mice environment. There are plenty of experience that explains a lot of things. But for the lamprey rearing system, this is kind of only laboratory. Our collaborator and us are the only laboratory that dealing with that. So we don't have so much experience, and we are doing this for maybe 3-4 years together. So it's a bit difficult to conclude what is the difference or what is the source of this variation. Maybe this could be the different year and different maturation status of the parents, or maybe the water kept there is slightly different temperature, or the food is slightly different or something like that. So there are a lot of factors that can be that can explain this variation. But if it's really the difference from the genetic background, like the hybrid from the other species or something like that, this would be very interesting. So first we have to clarify what makes this variation in a very accurate manner. Otherwise, I would say it's a bit dangerous if I just think, oh, this must be the genetic background and this can be explained by genetic experiment. This is a bit difficult to say. So maybe if we have more experience on this lamprey rearing system, maybe we can reach to some conclusion. So sorry for a bit ambiguous answer, but this is what I'm thinking right now. Thank you for the question.
Thank you very much. We have another question regarding hagfish. So I was reading one of the Thomas Boehm reviews and from 2012, and he says,
did you share it with people?
And I with some of them, I just completely forgot to put it on Twitter.
Yeah, very nice. So thank you very much for reading that. I'm very happy that you find it very kind of helpful. So.
Yes, it was very helpful for this presentation. So they say that in hagfish, they see agglutinin or production or antibody-like proteins production after infection only when they change temperature. So I don't know if the hagfish CD8-2 is dependent on temperature or
the stimulation dependent or not. Okay, so hagfish thing is kind of even more or even less studied. So far we don't have good method to keep hagfish in the laboratory environment. Do you know where the hagfish live? So they live in kind of deep in the bottom of the sea, around 200 meter bottom.
Okay.
So it's quite difficult to deal with them. But at least in the, in the experiment, they catch the wild hagfish, and even without any stimulation or something, they just have VLRB or VLRAC expression. And of course, if they inject some foreign red blood cell or something, then VLRB expression is hyper upregulated. So we are still not sure whether this is kind of like to have VLRB or the other VLRs in hagfish is a preset or like a stimulation dependent. But anyway, in the wild environment, they might have a lot of contacts to the pathogens. So there must be a lot of stimulation to generate more antibodies and more T-like cells and so on. So I think even without stimulation, VLRB can be observed in that species.
Okay, thank you. I don't know if you guys have any more questions. Just
thank you.
Yeah. Just
sorry, I have a question.
Yeah, please. Thank you.
Yes, okay. My name is Enrique. I'm from Mexico. Do you know which are the signals that induce the expression or the activity of the CD8-2?
Oh, this is very interesting question. So, yeah, this is really what I want to know, actually. So, yeah, I'm very impressed with all of your questions. This is very simple, but you gave me a lot of important questions. So, yeah, so what is the signal to start CD8-2 expression? So if I rephrase this question leads to the bigger question, how lamprey lymphocytes are developing. So differentiation of lymphocytes in lamprey. So this contains much bigger questions. So it's quite difficult. So I'm not sure where the hematopoietic cell of lamprey lies. Is it kidney? Is it tiflosol or somewhere else, like unknown small organs? Or so very basic information is missing in the lamprey immunology field. So maybe. So if you are kind of familiar with how this lineage was determined in the mouse or human system, this is really important to have a lot of infrastructure, like surface marker antibodies and like injection models and transplantation and which population can develop which lineages and so on. So this is like doing the same thing in a different species from almost zero. So from, as I said in the previous question, all we know about these lamprey lymphocyte lineages is VLRB is definitely VLRB cell lineages, and VLRA, C is kind of independent. So B and T is kind of separated, but even kind of differentiated cells, we are not so much sure whether VLRA or VLRC cells are completely independent lineages or not. So this is really interesting and deep question. Yeah, I would like to find, and sorry, the answer gets a bit longer, but short answer is, unfortunately, we don't have any information so far.
Thank you.
Thank you.
Let's see if we could, if there's anyone, if they want to ask any other question or.
Yeah, it's a really great opportunity. Thank you very much for everyone attending this conference and making so many interesting questions. I'm very happy to answer.
Yes. Yeah, and thank you very much.
Yeah,
you to have us.
Thank you very much.
Honor. Yes.
Yeah, so in the future, or hopefully in the near future, after this current situation gets a bit settled, I'm very much looking forward to meeting you guys in somewhere in the immunology conference or something. So if you find me, or please just feel free to contact me, and I'm very happy to take any answers or any questions regarding anything. So thank you very much.
Thank you, Ryo.
Yeah.
So I don't know if there's any more question. I want to ask all of you guys for attending and mostly to Ryo for having the time to
thank
join
you so much from my side. I
And
learned a lot.
We're really impressed with your work, and we want to ask if we could take a picture of the meeting. With, if you don't mind. And also, if you don't mind, we are planning to upload this conference on YouTube. So I don't know if you mind. Is there any problem if we upload the video?
It's okay. Yeah, if I said something wrong, just cut it out. No problem. Yeah, thank you.
Just a couple of seconds for if anyone wants to say something.
Thank you.
Please turn on your camera.
Sure.
Aranza. Okay. So it had Carlos, and we have Sven. I don't know if Raful or Alejandra, I think. Sven, if you could turn your camera. I don't think they have. So just okay, we have.
Can you turn on?
Yes.
Okay,
Okay.
perfect.
So just wait a second. That.
He's not. Okay. How about now?
Yeah. Oh well. No.
What? Come on.
No worries.
Well, maybe you could send us a picture, and I will put it there in the.
Let's do it like this.
Wait, just 1 second. Okay, 1, 2, 3. Okay, there you go. Thank you guys so much, Ryo. We want to invite you and your colleagues that next seminar.
Oh, great. Thank you.
We're gonna have. Let me show you this pamphlet.
Yeah. Also, thank you very much for making such a nice flyer. I'm surprised.
Thank you.
Yeah,
So-
you're so very well prepared.
So we're gonna have Andrés Hidalgo and José María Drover, which are researchers at the Fundación Centro Nacional de Investigaciones Cardiovasculares. Andrés Hidalgo has a lot of work with neutrophils, and he recently published Nature Immunology.
Now Andrés Hidalgo is in USA. Is in
Where?
Cold Spring Harbor.
Ah,
Yeah.
cool.
Now
Okay.
postdoc.
Well, anyway, Jose is in Cold Spring. Andres is a PI in Spain, and he's gonna join us for a discussion on neutrophils in this in our next session of Public Journal Club. So once again, Ryo, we thank you very much for joining us, and hope you take account our questions. Thank you so much.
Yeah, thank you so much. So all the
Thank you,
best for your studies and so on. Thank you very much for the organizing.
Thanks.
See you.
See you. Have a nice day.
next Saturday.
Bye bye.
See you. Thanks.
See you. Thank you very much, everyone.